Hello everyone, welcome to my channel. Today in this video I am going to discuss about cell organelles. What is cell organelle? The cell organelles have the same status in a cell as the organs have in the entire body of an animal or a plant performing specific function. Cell organelles are living parts. So whatever cell organelles are present in the animals, all are living and they are performing function as if the organs are performing functions in our body like heart, lungs, kidney, liver. All these kind of organs are present and they are having different functions and performing like that. All the organelles present in the cell are performing different functions. So we'll see what are the different cell organelles are present in the cell. First one is this, the outer membrane you see is a cell membrane. It is a cell membrane, and in plant cell one more membrane is present outside the cell membrane, that is called cell wall. So in this animal cell the outermost covering is the cell membrane, and in plant cell the outermost covering is the cell wall. Cell organelles are present in the cell are endoplasmic reticulum, ribosomes, nucleus. This is nucleus and the outer membrane is called nuclear membrane. This is endoplasmic reticulum. Ribosomes, these are ribosomes. They are present freely in the cytoplasm or they are present attached with the endoplasmic reticulum. Nucleolus, this part inside the nucleus, it is nucleolus. This outermost covering of the nucleus is called nuclear membrane. This is smooth endoplasmic reticulum. Why it is smooth? Because ribosomes are not present in this, that's why it is called smooth endoplasmic reticulum. This is nucleus. This is mitochondria. Vacuoles are present. Mitochondria is present. Centrosomes are present. Golgi apparatus, these are present. Lysosomes, this is lysosomes, and this fluid is present inside the cell is called cytoplasm. So today in this video I am going to discuss about endoplasmic reticulum. Endoplasmic reticulum, it is present in the cell. The structure of the endoplasmic reticulum, it is so fine that we cannot see without the microscope. When we are keeping the cell inside the microscope, under the electron microscope, then only we can see this endoplasmic reticulum. It is a fine structure that we can see it under the microscope. It is an irregular network. It is an irregular network of double membrane. This is double membrane distributed over the entire cytoplasm. This is a cell nucleus. So it will be attached from the nuclear membrane to the cell membrane. The inner end, this is the inner end is connected with, it is connected with the nuclear membrane, and its outer end is connected with the cell membrane. This is nuclear membrane. This is a nucleus, it is attached with it, and endoplasmic reticulum, it appears rough when the small granules called ribosomes are attached to it. It appears smooth when it is without the ribosomes. This is a nucleus and this is endoplasmic reticulum. When it is attached with the nuclear membrane, and you can see the small granules are attached with the endoplasmic reticulum. So when it is present, it is called rough endoplasmic reticulum, and when it is not present on the endoplasmic reticulum, it is called smooth endoplasmic reticulum. It does not work alone. The ER works closely with the Golgi apparatus. This is Golgi apparatus. This is Golgi apparatus and ribosomes. And this small dots you can see present on the endoplasmic reticulum is called ribosomes. Next point is endoplasmic reticulum looks different from cell to cell depending on the cell functions. So the structure of endoplasmic will look different depends upon the different cells. Components of endoplasmic reticulum. Components will be components of ER is that is cisternae, vesicles and tubules. Cisternae, these are long, flattened and unbranched unit along with the stacks. So in this diagram we can see what is cisternae. This is cisternae. This is cisternae. They are long, flattened and unbranched with units arranged in stacks. So these are the vesicles and tubules. These are vesicles and tubules in the endoplasmic reticulum. Vesicles, these are oval membrane bound structure. Tubules, these are irregular structures. Tubules and vesicles. Tubules they are oval membrane bound structures. Tubules these are irregular structure, mostly found in the smooth endoplasmic reticulum without ribosomes. Here these are the smooth endoplasmic reticulum. These are smooth endoplasmic reticulum. Next types of endoplasmic reticulum. First one is smooth endoplasmic reticulum, or you can say a granular endoplasmic reticulum. Endoplasmic reticulum. It has smooth membranes. It has smooth membrane without the ribosomes are not present. That's why it is called smooth endoplasmic reticulum. It acts as a storage organelles. So these organelles will store, and it helps in the carbohydrates, phospholipids, lipids, and helps in the storage and synthesis of steroids. So what it will do? It will help in the synthesis of it helps in the synthesis and storage of steroids, lipids, phospholipids. What is steroid? Steroid is other hormones are cyclical chemical compounds made up of rings of carbon atoms that plays an essential role in growth, development, energy, metabolism, etc. and reproduction. So this will help in the growth, development, energy, and it is mainly made up of vesicles and tubules. The synthesis of these steroids, lipids, phospholipids will take place in the smooth endoplasmic reticulum, and it helps in the synthesis of lipids, phospholipids, and these phospholipids and lipids helps in the formation of cell membrane. This cell membrane, it helps in the formation of cell membrane. Cell membrane is made up of phospholipids, bilayer phospholipids and proteins, and they are synthesized in the endoplasmic reticulum. Next is rough endoplasmic reticulum. Second one is rough endoplasmic reticulum. Why it is called rough endoplasmic reticulum? Because in the endoplasmic reticulum ribosomes are present. That's why it is called rough endoplasmic reticulum. And this rough endoplasmic reticulum is engaged in the protein synthesis. Rough endoplasmic reticulum is rough because ribosomes are present on the surface of the endoplasmic reticulum. So this helps in the synthesis of protein. These ribosomes will help in the synthesis of protein. It is mainly made up of cisternae. So this rough endoplasmic reticulum is mainly made up of cisternae. Tubules are very few. Now next we'll see the function of endoplasmic reticulum. So function what will be? Now next we will see the functions of endoplasmic reticulum. The first point is it provides channel for the transportation of materials between various regions of cytoplasm. So this is attached with the nuclear membrane and with the cell membrane. So it provides a channel to move the materials from various parts of the cytoplasm, and it transports material in and out of the cell. Second point is it's also function as a cytoplasmic framework, providing a surface for some biochemical activities in the cell. So it is providing a framework, surface for biochemical reactions. Third point, it also provides a support to the cell. It forms a supporting framework of the cell and also serves as a pathway for the distribution of materials from one part of the cell to the other part. Third point is smooth endoplasmic reticulum plays an important role in detoxifying many poisons and drugs and is also a site of lipid synthesis. So it helps in the detoxifying, means removing the toxic things from the cell, and it helps in that detoxifying many poison and drugs, and it is a site of lipid synthesis. Smooth endoplasmic reticulum helps in formation of lipids, phospholipids and steroids. Fourth point is rough endoplasmic reticulum is engaged in the synthesizing protein enzymes. So ribosomes are present on the rough endoplasmic reticulum. It helps in the synthesis of protein, and proteins are used for the growth and development of the cell. Fifth point is the proteins and lipids synthesized by rough endoplasmic reticulum and smooth endoplasmic reticulum respectively helps in building the cell membrane, which is known as membrane biogenesis. As we know that cell membrane is made up of phospholipids and proteins. So these are synthesized by this endoplasmic reticulum, and this is called formation of cell membrane. When it is damaged or required, this process is called biogenesis, membrane biogenesis. Next we will see ribosomes. Now ribosomes are the smallest and smallest cell organelle, and in this outer membrane is not present. It is a membraneless cell organelle. Membraneless cell organelle, and they are made up of RNA and proteins. Which helps in the formation of protein, or it is called the protein factory of the cell. These ribosomes are called the protein factory of the cell. Ribosomes are the protein builders or the protein synthesizers of the cell. These proteins might be used as enzymes or as a support for other cell functions. So proteins are required proteins are called the building block of our life. So protein is required for the growth and development, and proteins are synthesized by these ribosomes. These proteins might be used as enzymes or a support for other cell functions, or as support for cell functions. So ribosomes, we'll see where in the endoplasmic reticulum it is present, and these endoplasmic reticulum ribosomes are present, and some ribosomes are present, they are scattered in the cytoplasm. Ribosomes are found in many places around the cell. They are floating in the cytoplasm, and some are present in the endoplasmic reticulum. And these ribosomes helps in the synthesis of protein, and that will be used by the cell, or it will be used for exporting proteins to the other cells. The eukaryotic ribosomes are 80S while the prokaryotic ribosomes are 70S. I have discussed about eukaryotic cells and prokaryotic cells in the previous video. So eukaryotic cell will have 80S and prokaryotic cell will have 70S. Mostly prokaryotic cells are unicellular. These cells are unicellular. What is S? S is Sedberg unit, and S stands for sedimentation coefficient. What is sedimentation coefficient? It is measured as the density and size of the subunits of ribosomes. Eukaryotic cell will have 80S. These ribosomes have two subunits. Ribosomes are made up of two subunits, that is, the larger subunit is made up of 60S and the smaller subunit is made up of 40S. Why does 60S and 40S makes 80S? 80S ribosome is identified according to its sedimentation. What is sedimentation? Means it is the density of the ribosome. How does it get settled down? So since it is measured by the sedimentation time, it is not simply the sum of the subunits 60S and 40S. The 60S and 40S are two separate subunits that together sediments at 80S. I hope it is clear. Thank you. In the next video, I am going to explain about Golgi apparatus and mitochondria.